All right, ladies and gents, so today we're going to start talking about cells and really just kind of focusing in on their cellular structure. But before we can get into all of that, let's talk about the fact that in order for you to be able to visualize and see those cells are microscopic. They're not something that you can see, generally speaking, with your naked eye. So instead, that means we need some sort of tool in order to see those cell structures. So that's where those microscopes come in. Just a couple of terms that go along with those microscopes. If we talk about an image that is actually produced by a microscope, there is a term for it, and we call those a micrograph. Now, depending on what sort of question you're asking as a scientist, what sort of structures that you want to be able to see, microscopes have a very finite magnification and resolution to them. So you need to make sure that you're picking the right tool to answer those particular questions. So when we talk about magnification, we're just talking about how much bigger can I make the image compared to what it actually is? How much can I zoom in? The resolution, on the other hand, is the clarity. How clear of an image can I create when I zoom in? So anything, whether we're talking about a microscope, a camera, a telescope, everything is bound by this idea of magnification and resolution. So I can magnify things from here till the cows come home. But if I can magnify it, can I make it a well resolutioned image? Can I see it? Can I make it clearer? Because if I can't do both, then it really doesn't matter. Think about a picture on your cell phone. You can zoom in, you can zoom in, you can zoom in, but eventually you start losing the resolution. The images start going blurry and pixelated, and you lose the data. So it's not really helpful, even though you could continue zooming in. You have that capability. It's not actually valuable information. So depending on the organism you're studying, the questions you're asking, we really, as biologists, are bound to two major categories of microscopes. And those are your light microscopes and your electron microscopes. And choosing the proper tool to answer the questions is really important. So most of you guys have probably come across somewhere in your schooling, probably in middle school, these compound light microscopes. And they're compound microscopes because they have two lenses, okay? And you're looking through two lenses at a shot. That's where the compound piece comes from. So what does that actually do? Well, you've got some sort of light source in the bottom. You've got your object, your slide sitting here on the stage, and you're then viewing through an objective lens plus up here into the eyepiece where you actually put your eye to look through. Now, there are variations in terms of what these light microscopes are capable of doing. We'll kind of focus in on what we're capable of here at the high school with the microscopes that we generally have. And most of the microscopes we're going to use this year have three objective lenses. So we talk about a high objective, a low objective, and then the medium sits in between. So if I look at total magnification, high gets me 400 times its actual size, the medium gets me 100 times its actual size, the low gets me 40 times its actual size. Now you can have one additional lens in this system, which is actually higher than that high objective lens. And the microscopes here that the infectious disease students use, those go up that high. They've got what they call oil immersion lens. So I actually need oil because I can't look through the air to make them work. But they can go up to 1,000 times the actual size. But that's the top end of a light microscope. I can't go any higher than that. So there's benefits and drawbacks to this particular tool. Okay. Drawback is that I can only get 1,000 times the object's actual size. That just barely scratches the surface of being able to see the exterior structure of a bacterial cell. But the benefit is that I can look at living organisms. I can put a drop of pond water under this microscope and be able to see organisms floating around and interacting. That's not necessarily going to be the case when I get to those electron microscopes. So if I take a look, here's an image at the top end of what we could see with our microscopes that you guys will be using this year. And this is a paramecium, so this is something that you might see in a drop of pond water. And we can watch these guys swim around. They've got cilia, so these little hair-like projections that they use to swim. But here's a entire single-celled organism at 400 times its actual size. So you got a microscope review worksheet today. You guys are going to take some time today and go through and try to label the diagram, figure out what the function of those parts are, and we'll talk about those answers later. You will be responsible for knowing this for the test. But if you look, these are the basic parts of any light microscope. Now, some microscopes are more advanced, and so they have some additional pieces, but most traditional high school level compound light microscopes will have these pieces, and they'll all perform the same function. So we'll talk about that a little bit more in detail when we go over that review worksheet in a little bit. But let's move on and talk about, what if I need more magnification? All right, what if I need to see something more than 400 or more than 1,000 times its actual size? Then those light microscopes aren't going to do it for me. I need something that has a higher magnification, and we've topped out those light microscopes at 1,000 times its actual size. So now I need to go to an electron microscope. And just like there are some options with light microscopes, there's some options with electron microscopes as well. And these guys, I have two major categories of them. The transmission electron microscope is what we're looking at here, or this TEM. So it is a big machine. They usually kind of take over a small room in a university or a research lab to house these. They are pretty big. You need a computer system hooked up to them. And this big tube here is what's going to focus the beam of electrons. And in here is where you put your sample. Now, here's the thing with electron microscopes. I can only look at dead organisms, and that's because of the preparation and the way that the scope actually works. So with a transmission electron microscope, I'm going to be able to see internal structure. But in order to achieve that, I have to really thinly slice my specimen, which is clearly going to kill my organism in the process. So there's one thing. The other piece is that this little chamber here is a vacuum chamber. So I'm going to evacuate, I'm going to suck out all of the air so that I'm not hitting electrons off of air particles and only off of the specimen that I'm studying. Well, if I stick you in a vacuum, you're going to die. All right, so not really going to be good to view living things. So once I get to this type of magnification, the only thing I can really observe is structure. Light microscopes, if I can manage with that sort of magnification, I can see structure and potentially function. This one, we're going to have to make some hypotheses about the function because I can't actually observe it functioning under a microscope. So how does a transmission electron microscope actually work? Well, your really thinly sliced specimen goes in here. We send beams of electrons across that specimen, and there's a detection plate under here that's actually connected to your computer over here. And what that detection plate is looking for are, where does the electron actually hit and where does it not? So sort of think of it like lining up a bunch of students along the back wall of the school, right? And we just pelt them with paintballs, okay? And then you have everybody step away. What are you going to see? Well, you're going to be able to see the outline of where the paint didn't hit because it hit somebody's actual body. You'll see voids. And that's exactly what that detection plate is doing and the computer software is doing. It's taking that information of where does the electron hit, where does it leave a void, and then uses that information to actually build us an image. And this is a particular image that we might be able to obtain from a transmission electron microscope. Nowhere near the top end of what an electron microscope can do. We can talk in the millions in terms of how much magnification I can get out of it. But what we're looking at here is that same paramecium, all right, but I've zoomed into just under 15,000 times its actual size. So go back to that paramecium. Remember I said they've got these little cilia, these little hairs that are stuck into their cell membrane so that they can swim? That's what we're zooming in on. Here's a single one of those little cilia hairs. So incredibly more detailed image that I could never achieve with my light microscope. But The problem is I can't watch this structure actually function because I've got to kill the organism in order to see it. Now, transmission sees inside. Your scanning electron microscopes see the surface, which is also pretty cool. Still can only see dead organisms because, once again, all right, here's one of these microscopes. This is a vacuum chamber. I've got to suck it out of the air. But I also have to spray my specimens now with a really fine metal coating. If I started spraying you with metal, your body's not going to like that so much. And so the same thing happens here. But I now get an image of the surface, which I can't get with a transmission electron microscope, and I can't easily get with a light microscope either. That light has to pass through my specimen in order for me to see it in the light microscope. So all of a sudden we get a whole new vision of what life looks like at this scale. So still produces me images with a computer, so although they're not showing you the computer here, it's kind of sitting off to the side. Only major difference is that here is that vacuum chamber, my specimen sits on the inside, beam of electrons comes down, but now rather than your detection plate being underneath it, it's actually up here. And we're looking for those deflections. Okay, and where does that electron get bounced back to? And that's what the computer software is then going to take that information and produce us an image of. Okay, so here's my paramecium, not super high magnification compared to what a light microscope can do. Remember, a light microscope can top off at 1,000 times its size. This one's only 1,600 times. But look how fuzzy he is. You would never be able to see this with a light microscope, right? Go back to that light microscope picture. I can't see that because I'm looking directly through it. All right, they're clear organisms. So I can see them. They're transparent. Okay, we'd never be able to know that that's what a paramecium looked like on the outside without having the scanning electron microscope to scan over the top of that cell and be able to see that it looks all fuzzy like that. Now, we do actually have one other option. So what if I need to see something at a really high magnification, but I really need to have the potential to see it alive? Well, that's where these new scanning tunneling microscopes come in. And I say new in terms of the last few decades. This isn't something that has been highly put out there more recently. But what they do is you'll stick your specimen in these tubes, and they actually have needle-like probes that are going to go and measure the electrons that are leaking from the surface of that object. So any of us are slowly decaying. We talk about it as like radioactive decay and that kind of thing. Similar to that. And so those electrons are getting bounced off, and these scanning tunneling microscopes are able to read those electrical differentials and be able to then take that information to a computer, and that computer then can produce an image. So really high magnification, even more than that, like approximately 2 to 300 or 2 to 3 million times its size. Now I can get down to an atom, which is really tiny. But the cool thing is now it allows me to see both living or deceased organisms. So I get the potential to not only see really high magnification, but now I can actually see how that thing works, not just what the structure looks like.